Welcome, everybody. Today I'm speaking with Dr. Monica Justice, who is calling in today from Houston. She is a professor in the Department of Molecular and Human Genetics at Baylor. Welcome, Dr. Justice. I'm going to try to remember to say Dr. Justice and not Monica.
Thank you.
So she is a world-renowned expert in mouse genetics, and because the human genome and the mouse genome are surprisingly similar, she uses mouse genetics as a tool to better understand the function of mammalian genes and to try to gain insight into human disease. Do you have anything to add to that about your background and what it is that you do?
I think that was a very nice description.
Okay.
That was fine.
So we're talking today because your paper was just accepted at Nature Genetics. Congratulations.
Thank you.
And the title of the paper is A Suppressor Screen of Mouse MECP2 Implicates Cholesterol Metabolism in Rett Syndrome. So that will give our viewers a little bit of insight into what it is that we're going to be discussing today. So being at Baylor, and being in the same department as Dr. Zogby, certainly you knew about Rett syndrome probably years before you started this project. But this is the first time you actually worked on Rett, correct?
That's true. I've been on some of Dr. Zogby's graduate student committees, and I was very aware of the work. The gene was actually discovered right around the time I moved to Baylor 15 years ago.
Yes, time does fly.
Yes, it does.
Dr. Justice, why don't you start us off by explaining the concepts of modifiers and why they're important in the context of trying to treat disease?
Well, let's talk about what a modifier is. Now, you can imagine it's just what it says. It can change or modify something. So I like to use the analogy of, for instance, something like hair color. We might want to make our hair lighter or darker. We might want to highlight it. But we can change it from its original color. We could even make it completely different. Well, the same thing can happen with a disease gene. The disease gene can have a certain outcome and symptoms, or it can be affected by the presence of other mutations in the genome that modify its outcome, so it may be more severe, or it may be less severe, or it may even have a completely different clinical phenotype. And so this is a common concept in human genetics and in mouse in genetics, period. And we felt that this could be exploited in Rett syndrome, for example.
Can you give us some examples of some well-known modifiers that have been identified in other diseases?
Yeah, there are actually a couple of pretty famous ones. One, probably relevant to neurological diseases, is that spinal muscular atrophy is modified by mutations in a second gene called plastin. And they first discovered this in humans, where even related individuals with the disease could have hugely different phenotypic outcomes, very severe disease or almost no disease. Another, to me, classical example is a predisposition to gastrointestinal cancer caused by mutations in a gene called adenomatous polyposis coli. And I teach about this gene a lot because this gene causes a lot of polyps in your intestine, and these polyps predispose to GI cancer. However, mouse geneticists isolated a suppressor based on just crossing with inbred strains that reduced the number of polyps and therefore reduced the incidence of GI cancer in these mice.
Okay, so they really haven't. They can have some very dramatic effects on how a disease manifests itself.
Huge effects, yes.
Okay. So now that we know what modifiers are, can you tell us the goal of your Rett syndrome project and the methodology, how the experiment is done?
Well, when we approached this project, actually after you had contacted me, I really felt that with Rett syndrome and with the male mice being a model, that our best approach would really be to see if we could suppress disease symptoms and therefore increase longevity of the mice, since all of these male mice would die if they carried the mutation. So our approach was really one that we decided should be an unbiased approach. So one approach I could have taken, for instance, was to take an existing mouse mutation that I thought might suppress symptoms in the male mice and breed it with those male mice and then see if I was correct. But I really don't think I'm that smart, and I really felt that the best way to do this was to let the mouse tell us what's important. So, in other words, I would use a chemical mutagen that I'm kind of well known for called ethyl nitrosourea, and it's a random mutagen that creates all types of mutations. There's no bias, and we simply use that mutagen and see if we can isolate a mutation that will improve the symptoms of disease in the male mice.
So you give the drug to male mice, you breed them with the female Rett mice, you watch the offspring. And
Yes.
the offspring will have no MECP2 from their mothers, and they will have inherited a random mutation from the father who got it because of the drug. And then you watch the offsprings, and whichever ones do better than your typical Rett mice, you then go back and try to find where that random mutation was.
Yes, and this requires lots and lots of mouse breeding. So in other words, we look for this amelioration of symptoms in the very first generation of breeding from the MECP2 female and the ENU male. And then, once we see something, we have to breed that. We have to ensure that it's transmitted genetically. We then have to segregate other loci away and come up with a locus that confers suppression. So it's a lot of mouse breeding.
A lot of mouse breeding. You brought up a good point about the screen being unbiased. So we're not going into it with any preconceived notions of what the pathways might be that interact with MECP2. And there's a lot of value in that because,
Yes.
like you said, we may not be smart enough to figure out which genes MECP2 is interacting with.
Exactly.
So your paper represents the first time that a modifier screen in mouse has actually been used to detect a potential treatment in disease. Can you elaborate on that?
That is correct. So when we decided to do a suppressor screen, although I wasn't sure it would even work, I thought that, okay, so MECP2 binds DNA. We all know that. It supposedly interacts with other transcription factors that modify chromatin, and working with DNA in a therapeutic sense is actually very, very difficult. I think everyone knows that. So in the back of my mind, I thought, well, by doing this screen, we might be able to identify an alternate pathway that doesn't involve DNA, but that maybe it involves how or what causes some of the pathology of disease, and that pathway might be therapeutically targetable. And so we really hoped that we would find such a thing, although I wasn't sure that we could with a DNA binding molecule. And in this case, it was the first suppressor screen that was really designed to go after therapeutics, and in fact, it has led to a potential therapeutic target.
So you are currently 15% of the way through the screen. There's still a lot of screening that needs to happen, but with 15% of it under your belt, you've identified 5 modifiers that are written about in the paper. Can you tell us about them?
Yes, I can tell you a little bit about them. I should back up and say that in the paper we really focus on the one modifier that we identified that affects cholesterol metabolism because it's the one that led to therapeutics. However, I'm going to tell you that the others that we have found are not genes that we would have predicted. For instance, they're not ones that I would have guessed might be found to modify MECP2. However, they make a lot of sense. They're all involved in remodeling chromatin, and so that's perhaps what we might have expected from a modifier screen, and yet it's not clear that any of them will be therapeutically targetable. However, I do think they're going to teach us a little bit about the mechanism of action of MECP2, sort of how the disease occurs in the long run. But our real focus here was on this therapeutic pathway. So we jumped on it right away and decided to go that route.
Okay, so this is a gene called Squalene epoxidase. It's involved in cholesterol metabolism. This is not the first time that cholesterol has been implicated in neurological disease. Can you tell us a bit about what the relationship is, cholesterol in the brain?
Okay, yeah. So I think most of us think of cholesterol as something that we have to avoid in our diets or go on statins for because it's too high in our bodies. But, however, cholesterol is an essential molecule that's required for nearly every cell in your body. And one of the things we never think about is the fact that cholesterol can't cross the blood-brain barrier. Your brain needs tons of cholesterol. It uses it to myelinate all of your axons. It requires it for lipid rafts. It is involved in neuromodulators. It is involved in dendrite remodeling, which is one of the neurological defects in Rett syndrome. But your brain has to manufacture every bit of cholesterol that it gets inside you. In other words, there, in the brain. Now, the brain can talk to the rest of the body by by hydroxylating cholesterol and then turning it over into the circulation so it can be eliminated. And your brain constantly has to turn over this cholesterol. It likes for it to be fresh. In other words, it likes for it to be new. So cholesterol turnover in the brain is very, very important. We usually don't think about things like this, I don't think, and I don't think we thought about it in Rett syndrome. However, it is an emerging concept in neurological diseases, as you have mentioned. For instance, it is known to be involved in a number of very severe early birth defect disorders, such as desmoid sclerosis, Smith-Lemli-Opitz disease, but also now in things like Lou Gehrig's disease, Alzheimer's disease, even Huntington disease. And so I think this is an emerging concept. I must say that I think cholesterol metabolism is very much easier to study in, for instance, your blood and your liver than it is in your brain, because your brain is so complex. Okay.
So can you tell us what this gene does?
Yes. So squalene epoxidase is its nomenclature might say, it adds an oxygen atom to a cholesterol precursor that's called squalene. Now, most of us, when we think about cholesterol, we think about HMG-CoA reductase, because that is the enzyme that statins target. HMG-CoA reductase is way up here in the pathway towards generating cholesterol, and there's a whole part of the pathway that has a lot of molecules in it that are required for a lot of other processes, including, how our muscles work. But squalene is the first immediate precursor to the committed cholesterol pathway. And what happens is that cholesterol feeds back to both squalene epoxidase and to HMG-CoA reductase to regulate how much cholesterol is produced and how well this whole pathway, how much it synthesizes.
Okay. So mutations in this gene is what you found in your screen. How did you correlate that mutation in this gene to, let's try statins? What's the correlation there?
Well, the correlation was that the mutation itself is a loss of function mutation in this enzyme. So we then we realized that essentially a loss of function mutation. Now remember, it's acting as a dominant mutation or as a hemizygous mutation, is going to act to downregulate the cholesterol synthesis pathway. So we thought, well, another molecule that would downregulate this pathway are statins. So let's go see if statins, treating the mice with statins, downregulating cholesterol synthesis in these mice would actually improve symptoms. So that's what we did. And it improved symptoms.
Can you tell us a little bit about that? What improved, what didn't improve?
Yes. Now, as you can imagine from any modifier screen, and especially a gene as complex as MECP2, we did not get complete rescue of all symptoms. There could be issues partially in dosing, in how the statin acts, but also just the fact that it probably doesn't rescue all the symptoms. But what we did find is that it tremendously rescued motor symptoms. So the ability of the mice simply to move around, their general health, they're much healthier. We actually have videos that are available with our paper online where you can go and you can see how much healthier the mice are, how much better they move. But we also use some behavior assays such as open field activity and a motor performance assay called a rotarod, in order to make sure that what we were observing was actually quantifiable. This rescue of, I'm not going to say rescue, I shouldn't say rescue actually, this improvement of some of the symptoms also led to increased longevity in the male mice. We were not sure that this would show symptoms, improvement of symptoms in female mice. However, we wanted to also test female mice with the statin drugs to determine if this would have any relevance to, for instance, females affected with Rett syndrome. So we also did our studies in female mice and found that the statin treatment also improved the same symptoms in female mice as it improved in male mice.
Did it matter when you give the treatment?
That is an excellent question. Our treatment strategy was primarily focused on just one age, and that was we started treating the mice very early in their development of symptoms. So the mice usually have the earliest onset of symptoms around 4 weeks of age.
This is the male mice.
The male mice, and we started treating them at 5 weeks of age, right after they started to show symptoms. We then treated them actually for a long time because some of the mice lived very long, and we treated them with a very low dose of statin. So our statin drug treatment, actually the protocol we use now is administration of statins twice a week at a fairly low dose, lower than you would use to lower cholesterol in your body. And in the female mice, we started treating them at 6 weeks, and we usually ended the treatment strategy at 8 months of age because that's when females are usually very affected, and we just decided to end the study. So you can see also when I'm talking about treating these mice for 8 months to a year, this was a very long study. This is probably partially why our paper is coming out nearly 7 years after the screen started.
Right. So, testing symptomatic females to see whether symptoms are reversed is. Yeah. Are there plans to do that?
We have just started those studies. So one thing we would like to do is to take female mice that are highly symptomatic and that have developed some lipid accumulation that we have found happens in them, and see if we can improve symptoms. However, these studies have just started. We have a little bit of preliminary data that suggests that this does not help the mice as much as, for instance, starting a treatment at a younger age. However, it opens up the possibility that there could be other pathways within the cholesterol synthesis or lipid metabolism pathway that could be targetable to help improve other females. So we're testing, the older, more affected females. So we're actually in the process of testing some of those other things now.
Right, and there may be drugs that you can test in combination. You may have to test at different doses. So it seems like a simple concept to test, but actually, when you think about it, it's it's laborious.
Yeah, to do this in a complete way actually requires multiple doses. It requires fairly large cohorts of mice. It requires injections and observations and assessments, behavior assessments, over a long time period. And it's just a matter of how much we can do and how much we can handle. Yeah. Also, just to do the female study. So the female study that we did sort of preliminarily, we took older females, but we hadn't saved wild type ones as our controls. Mm-hmm. So it was we realized it was kind of a flawed study, and that's why it's not in our paper. Because we didn't have wild-type controls to compare with the MECP2 females. What we've done now is we have those controls and those older females that we're treating, and we feel it's a much better study than anything we've done before.
You also found some evidence of fatty liver disease in the mice.
Yes, we did. Now, lipid metabolism is a very complex event. I should say that each of us probably metabolizes lipids differently. My husband has very high cholesterol. It runs in his family. Mine does not. I seem to be fine. But you can imagine that from individual to individual, how your body manages cholesterol is different. So our screen and our initial assessment of cholesterol took place in a genetic background of mice that is very fixed. It's called an inbred strain, and that strain is called 129. Now, on this strain background, the mice develop very severe fatty liver. We feel that this is due to other metabolic modifiers in this strain background that allow lipids to accumulate to very high levels in the liver. And this is very bad for the mice. We have examined a separate strain background called C57 Black 6, where the lipids, the lipid perturbations in the brain are the same, and lipids do accumulate in the liver, but not at such high of levels. And so we feel that there is probably going to be variance in whether fatty liver might develop in different genetic backgrounds.
Okay. I mean, fatty livers is not something that one hears in Rett, but of course, have people looked? This is not something that probably most clinicians would look at. So,
my impression of the disorder is that it is primarily thought of to be a neurological disease. However, I am a mouse geneticist who works with whole animals, and I know that when you have perturbations in the brain and you have the right systemic components, that something that perturbs the brain can affect the entire system. And I'm a bit concerned that those systemic components may have been overlooked in Rett syndrome. I don't know. This is a mouse study, but I'm sticking my neck out.
The great thing about having your paper out there with these facts is it'll draw attention to the liver, and hopefully, clinicians will start looking.
And we'll see. We'll see if, this is just a difference between mice and humans, or if indeed this could be an issue in some patients.
Yeah. So has this, has your data kind of forced you to look at Rett syndrome differently? Not merely as a neural. I mean, do you think of it now more as a metabolic disorder?
Well,
in addition to a neurological disorder.
Let me tell you that I believe that Rett syndrome has a metabolic component. May I say that?
Of course.
And I think based on what we've seen systemically, there are probably going to be degrees of variation in how that systemic component is affected. And that, think about metabolism. My goodness, there are so many things that modify it. There could be a huge degree of differences in the patients, in sort of what is the ultimate phenotypic outcome of disease, or the symptomatic outcome.
So once you started telling me about your data, you got connected with Dr. Sasha Dukic, who reached out to you. I think she had learned about your data at a meeting, and we provided some funding to her to be able to hire a research assistant who was able to mine the database and go back and look at lipid data, cholesterol data in patients with Rett syndrome, and also order new data on new patients. And basically what Dr. Dukic found was that young children with Rett, about 50% of them have high cholesterol. And then as the children age, that cholesterol comes down and normalizes. We don't yet know why.
Yes.
But that's quite interesting.
Yes, and really, although this was a retrospective study and it wasn't really well controlled by diet and background, it's very consistent with our mouse data. So, for instance, we found that in this one strain that develops horrible fatty liver, we found very elevated cholesterol and triglycerides in the blood of these mice. However, in the other strain where the lipids in the liver don't get quite as high, we did not find elevated cholesterol and triglycerides. So we think that there's probably going to be a variation in the symptoms that are manifested, and that perhaps high lipids in the serum could be a biomarker in some patients and may actually be an indication for instance, for statin treatment in those patients.
Right. So Dr. Dukic has designed a trial testing a statin, and we hope to get that up and going within the next year or so. I do feel compelled to caution parents. I don't think they should go out and talk their pediatrician into putting their children on a statin now. Do you want to comment on that?
Well, this is actually one of our concerns in publishing this story, and why we were so careful in the way we've reported it. We've tried to be very clear on differences in mouse strain backgrounds, is because we really don't think this is going to be a fix-all. We also don't want people to think that just going on statins is going to fix it. There have to be good clinical trials that show us who might respond, or if patients will respond, and also what the appropriate dosage is. This could be very dangerous, and we're actually worried about people getting a hold of statins and self-dosing, for example. Yeah. That's just not a good idea. We found if we treat the mice with a really high dose of statins, they get worse.
Yeah.
So I think there's a lot of care that has to go into this.
Yeah. And there's not a lot of data out there about statins in typical kids with high cholesterol. It's just not a medication that's used in kids. So, a lot of care has to be taken. So the right thing to do is to wait for the trial. Let's do it the right way. Let's get the results. And if it does work, then we would have that information to move forward.
That's correct.
So in terms of next steps for you, obviously, is to continue the screen. We committed funding to you at the end of last year to keep the screen going, and it's been so far a $1.5 million investment from RSRT. We think it's an important project to keep going. From our perspective, we'd like to see the full list of modifiers, which we can then prioritize and see which ones are in pathways that are druggable, like the statin ones. Some will, some won't.
Yes.
But until we have that full list, you can't really prioritize and chase down the ones that are most tractable.
That's right. I think now that we have shown that this approach even works, that what we're going to do now is just dive in and try to identify all the potential pathways and see if there are any other targetable pathways. I should mention that one of the things that makes this approach possible right now is the ability to do DNA capture and massively parallel sequencing. In the past, I don't think this type of project would have been approachable. And now the technologies are allowing us to do this so rapidly that it almost makes it an easy project. In fact, technologies have changed so much since when we first started the very first screen that we can do this about 5 times faster than we could then.
Yeah, that's been exciting for me to see that, because this project really lets you see how technology changes in real time. Because by the time you're writing a progress report, things have changed, and the cost. So I was saying it's been exciting to see the technology improvements actually impact this project on a real time basis.
Exciting for me too.
Okay, so I feel compelled, as I always do, to thank our donors who may be listening. These are the types of projects that you help to fund. So we thank you for that, and we're not done with funding for this project. We've committed funding to get to the 50% mark, and because the cost of sequencing is coming down at the 50% mark, Dr. Justice is going to submit another proposal to us to get us to the 100% mark. And what is your timeline for that? You think you may have the whole list of modifiers by when?
I'm hoping that we would have this entire 100% project completed within 4 years. That's my goal.
Okay.
And actually, this initial goal went very quickly. We actually did the screening over a 2-year period to find the 5 modifiers. And the remaining time has really been spent trying to show what's going on with this squalene epoxidase modifier, determine what's happening with cholesterol biosynthesis in these mice, and then try to get our paper published.
Yeah,
so we're going to try not to be so focused on that in the next screen and just kind of...
Just do the screen.
Just do the screen and find the genes.
And hopefully, technology will continue to impact your project, and you'll be able to go even faster. So thank you very much for taking the time to speak with us and explain to our viewers what your project. And congratulations again on your paper. And we look forward to bringing more updated information to our viewers as your project continues. Thank you.
Thank you, Monica. And I want to thank all the donors as well, because if you hadn't believed in this project and supported it, it wouldn't have happened. It was a very high-risk project. I'm not sure it's considered so high-risk anymore, but it worked, and I think we're really going to go somewhere with the next screen. So congratulations to you, too.
Thank you. All right, bye-bye.